this is Miss Litton, and this is 7th period. Say hi.
Hi.
And we are going through the Big Idea reviews. We already did Big Idea 1, which was about what? Evolution. Okay, now, Big Idea 2, biological systems utilize energy and molecular building blocks to grow, reproduce. We have already done we've done Big Idea 2A, B, and C, and now we are on 2D, growth and homeostasis of a biological system are influenced by changes in the system's environment. So let's look at the response to the environment. We need to understand this. Do you remember this? Population is a group of what? Same species. And what is a community? Several different species. And what is an ecosystem? With the abiotic factors like the environment. And then biomes, remember we studied the different biomes? Remember, like if I said desert biome, what kind of flora would you see there? There's flora and fauna, right? What's flora? Plants. So you would see a cactus. What kind of fauna would you see there? Lizards. Does it rain a lot or rain a little? Little. What kind of temperatures during the day? Hot. And night? Cold. Right. So the climate is going to dictate the flora and fauna of that area. What are some other things that could affect climate besides where you are on the equator? Altitude, right? What else? Altitude and latitude. What else could affect it? Landfall, terrestrial structures like mountains, or if you're near what? Body of water. Good job. And then we looked at biospheres, and what are biospheres? All living things live in the biosphere. Okay, good. So then we looked at how the environment can push organisms. Then those, right, because you would be adapted for that particular environment. But in turn then, those organisms could have an influence back on their environment. When we talked about succession, you could have a small shallow body of water which could have cattails along the edges of it. And that's adaptive to have a cattail in that shallow body of water. But as those cattails die and they fill in the water with their dead debris, right, their detritus, then it can change it and make the body of water even more shallow until you have grasses in a meadow. And as those grasses grow, then maybe you have more soil as they die, and then you could have small shrubs and then what? Larger shrubs. So the environment can influence who can grow there, right, who would be adaptive and fit. But so too, that organism can influence their environment. How could lichens influence an environment? Make soil by doing what? Breaking down the rocks. Okay? Who probably influences the environment the fastest? Humans. You know what I heard today? Heard on the news today that Mr. Pompeo said? He said that we can now, because of the melting glaciers in Antarctica, that can open up trade routes for increasing trade. Please tell me that you could write a whole essay on how that would not be a positive thing.
I could write it in Spanish.
Yes, you could, because you passed your Spanish AP test. That was literally terrifying for me to hear. You understand that the melting glaciers is a bad thing, right? Yeah. Okay. All right. I'm just checking. Looking at this glacier here. You know the difference between abiotic and abiotic? Abiotic and abiotic. You know the difference biotic? We talked about predator-prey, right? Whoever is the oldest bio buddy that you're sitting next to, why don't you talk about how the predator and prey can influence each other and their numbers, and use this diagram right here. And do you remember seeing this on a practice AP exam?
Yes.
Okay, go ahead, talk.
So, as predators, so you have predators and prey. So they go down the day and then they go up and they crush the why are you here?
Okay, riddle me this. Could something else influence prey numbers besides the number of predators? Yes, like what? Rainfall. Lack of rainfall, right, would mean not enough things, right, to sustain them. Exactly. Okay. Next, food chains. You might notice in this environment, we've had such high rainfalls that we're going to have more plants, which mean more seeds. So you might notice more bunnies and things running around in your yard. More bunnies could be more food for things like what? Coyotes, maybe snakes. So those numbers could go up, right? Because remember how we talked about how energy flows and nutrients, what? Cycle. Yeah. So an increase in one can trigger another one. Now a food chain, we could follow a simple food chain, but what's even better than a food chain? Because it shows what? All the interconnections. Good. All right, and so this would be more likely a food web. Okay, when we talked about ecological pyramids, we would stack them up. We talked about you could do a pyramid of numbers. There are more blades of grass, right, than there are, let's say, zebras that are eating the grass. But pyramid of numbers doesn't always work. When does the pyramid of numbers not work? Give me an example. A tree with a bunch of monkeys on it. There you go. Or a bunch of insects or ants on it. So then we said not just the pyramid of numbers, but we not just maybe a pyramid of even biomass, like how big something is. Do you remember what the pyramid we said would be best? Pyramid of energy. How much energy is each level? And what did we say? How much can get passed on? 10%. About 10% gets passed on. Okay, good job. I liked this because it pulled in a couple of different things. This document, if you grabbed it, if you can kind of follow it through, can you see it all right?
Yeah.
Okay, youngest bio buddy, take the lead. Start up in the top. Follow this flow chart through. Use it as a review. Are you good? Are you spent? Can you see it? I just thought it'd be better if you talked it through than I talked it through.
Wait 1 second.
Okay.
Okay, gracias.
Got it?
Yeah.
All right. Next, homeostatic evolution. Oldest bio buddy. Why don't you explain this diagram right here? Go ahead, oldest bio buddy. Go quick.
Okay, so you have your population and environment. So the environment is worse than
Because
in the environment, then there will be population
All right, so the environment is going to dictate who's most fit, right? Those survivors in that population, they could be whoever's most fit, or you could have mutations or disturbances that could influence that. Those are the ones that reproduce, so it reinforces some trait, maybe a larger beak, or maybe more webbing in your feet, whatever it might be. Next, we're talking about all different things in our environment that can influence blood pressure. What can influence our blood pressure? Amount of salt is in our diet. What else could influence it? Amount of water, right? Stress. These are all things that influence our blood pressure. Hello, kidney. So remember we talked about skin adaptations. What are some adaptations for responding to your environment? What?
like sensory.
Oh, you could have sensors so you know if it's hot or cold. What? Taste, right? Don't just think about us. Think about all organisms.
Barriers.
Barriers that we have, protective barriers in our skin. You could have hair, you could have fat, right, to help you, spines, you could have poisons, you could have toxins, right? Those are all things that help you respond to your particular environment. So don't limit it just to yourself, right? When we talked about fish, whether they were saltwater fish or freshwater fish, they had to have different adaptations in their gills, right? Because if you're a freshwater fish, so just imagine you're a fish in fresh water, okay? Probably your body is what? High pertonic, and the fresh water is hypotonic. Water must flow. So a lot of water is going to flow into you in order for you to maintain your homeostasis in your environment. Are you going to pee a lot or pee a little? You're going to pee a lot. What might you lose? Salts. You might lose salts to your environment. Whereas if you are a saltwater fish, right? If you're a saltwater fish, you are probably what to your environment? Hypotonic, and you're going to be afraid that you're going to lose water to your environment, right? And have salts come in. So you always, and really good osmoregulators, ones you would find like in an estuary, where you could be bathed with fresh water or salt water, or if you're a salmon, where you move from freshwater to saltwater back into freshwater, then you have to be able to adapt to those different environments. We looked at things like chondriichthyes. Remember, chondriichthyes keep a real high level of urine in their blood, higher than we would normally keep, where they don't excrete it out through their kidneys, but they would keep it so that it helps them to be isotonic with the saltwater environment. Those are all ways. So if you ever get a question on dealing with your environment, those are all different things you can pull in for that, right? Okay. Oh yeah, talked about salmon. Oh, we did that. Can I move on?
Yeah.
Okay, because we talked about it. We talked about that. We know for terrestrial we tend to lose water to our environment. Sorry. Just went out. Water in, water out. I didn't even look at this presentation. I was just trying to think of things that would have to do with it. I think we looked at this in one other review, did we not? Yeah. Okay. All right. I'm glad I have a salt gland. Okay. Excretion. Yay. So we can look at several different adaptations. They are flame cells in planaria. Remember, they look because they wave. They're like cilia. That's why. And they usually are getting rid of excess water. Nephridia and earthworms are very similar to our nephrons because you have a blood flow that's going past a urine flow, right? And sometimes countercurrent, so you can pick up whatever you need to pick up or eliminate that waste. We talked about the need to deal with nitrogenous waste. Where do a lot of our nitrogenous waste come from? That's right, proteins. Because you're like so tired. You're like, don't be misled. Proteins, remember the structure of proteins?
Amino acid.
Yeah, remember amino acids, and amino acids have what?
R group.
What is that structure?
They have an amino group, an R group, carboxyl group.
Do you remember that? Yeah. And this hydrogen tends to come over here, and if it does, then this would be what? NH3. Remember, deamination for us occurs in our liver, and it's our liver who will convert it to urea, which requires energy, but then in excretion you need less water. What's something that requires more water but less energy? Ammonia. What requires the most energy and the least water? Uric acid, which is really good if you lay eggs, because the nitrogenous waste that you make as you're using up the yolk, right? As you're developing, that nitrogenous waste can be a solid, a precipitate out. Okay. Oh, look, and here's that picture. Yeah, remember all of that. Kangaroo rats get all the water they need from their metabolism. How can that be? Say it again? They don't drink water, but how can it come from their metabolism?
They're really good
Cellular respiration, what step?
No.
Who catches electrons at the bottom? Oxygen forming water. So by metabolizing their grains, their sugars, right, then they generate water, and that's the water that all that they need in order to maintain homeostasis. Okay. Yeah, watering holes you have to drink. Drink water. Okay. All right. Malpighian tubules. These kind of organisms have an open or closed circulatory system. Open. So their Malpighian tubules extend out into their coelomic fluids, and they can pick up whatever waste they want or leave those fluids in their body. We talked about this. You eat proteins. Your liver will convert it to your urea, and then your kidneys will filter it out, and then you will pee fire. Remember that? Okay. There are nephrons. This would be a great one to talk about maintaining homeostasis and the process of it. You could talk about countercurrent, right? Concentration gradients. You could talk about how, let me see if I put one in here. Did I? Yes. Okay, so remember at the glomerulus, that hydrostatic pressure, the force on the blood due to the heart, forces all fluid out into the glomerular capsule, good and bad, right? Like dumping out a purse. Remember, there's things we want to recover. So as it moves through the proximal convoluted tubule, about 67% of the salt is recovered. Who follows it? Water. Is that enough? No. Could that kill us if we lost that? Yes. Okay, and we also actively transport sugar and recover sugar. Then as it goes down the descending loop, what comes out?
Water.
And only water, because how is that set up? Because the
Ascending
ascending
loop,
loop,
salt.
only salt. Passive and active transport of salt, which makes this environment around the loop of the nephron or the loop of Henle to be very, very hypertonic. So water wants to come out at your descending loop and also once again at your collecting. And at your distal convoluted tubule, it's salt and water again, but that's also where you do tubular secretion. That is where you actively transport things out of your blood and into your waste tube, right? Into the distal convoluted tubule. And then the rest comes. What controls this collecting duct right here? ADH. Who secretes ADH? Posterior pituitary, right? And what does ADH do to the collecting duct?
It makes it absorb more water.
Makes it absorb more water out of the collecting duct and recover it into your blood, which could increase your blood volume, which it could increase your blood pressure. Good. All right. Oh, we just talked about that. We're on top of things. Oh, let's look at this. Do you remember this?
Oh, yeah.
Yeah, the liver secretes what?
No, wait. No.
Angiotensinogen, right? And we talked about renin making it go from and this is from the afferent arteriole. Anyway, angiotensin goes to angiotensin 1. ACE from the lungs makes it go to angiotensin 2, which makes the adrenal cortex secrete aldosterone. Aldosterone is one of the mineral corticoids, one of the steroid hormones, which helps with reabsorption of water, salts, and then water. Okay, look at that diagram. That is so good. You could have a whole essay on that just for your AP exam. You could totally nail it too. Okay, so renin, when there's low blood pressure, okay, so renin, you will have ultimately what we just saw here, okay, you will have this whole process take place. If you need the opposite of that, ANH from your heart will counteract renin, and it also counteracts ADH that we talked about earlier. If your blood pressure is too high, you remember all that, yes? Okay. Well, I know, but I just want you to move off that slide. And then we also talked about how the kidneys, besides blood pressure, we also talked about that it impacts you get rid of urea, and we talked about pH. We also talked about calcium. I guess I didn't put it in here. Calcium reabsorption as well. This hypothalamus pituitary thyroid connection, we reviewed that last class, right? What does the hypothalamus secrete? Thyroid releasing R4S, okay? Which makes the anterior pituitary secrete thyroid, which makes the neurothyroid gland secrete more thyroid hormone, which if the levels get high enough, it exerts what? On the hypothalamus and the pituitary. Okay, all helping to maintain homeostasis. Yes?
So what role does pH play with the kidneys?
Oh, that part is just that it can secrete bicarbonate or hydrogen ions to keep your pH just right. And remember, we want our pH right because our proteins, and we have blood proteins, right, and lipoproteins and things in there in order for them to have their shape. Yeah. Okay. Thyroid, besides thyroid hormone, we have calcitonin from your thyroid gland, which does what? Lowers your blood calcium. Who's counteractive to that? Parathyroid, which elevates it. Those are called contrary hormones. Okay, lymphatic system. This is fun. Okay, we totally remember that. The organs of your lymphatic system, we talked about B and T cells already. B cells are called B cells because they mature in the bone marrow. T cells are born in the bone marrow, but they mature in the Who does cell-to-cell combat? T cells. T cells doing cell-to-cell combat. Fist, right? And then B cells release what? Antibodies who can travel in the plasma and mark a foreign antigen for destruction. T cells. Who are you safe? Like, I don't need to worry about a T cell. I have
MHC1.
MHC2. I will help you. You display an antigen on an MHC1, you are done. Okay? How are lymph nodes and spleens similar?
They filter.
They filter, right? Spleens filter your blood, and lymph nodes filter your tissue fluid. Remember that part? Okay. We talked about barriers to keep things that are in our environment out of our body, right? You're surrounded by bacteria. Some of you genetically engineered some bacteria today. And so you're surrounded, you want to keep those pathogens out. Low pH of your stomach, oil on your skin, secretions, enzymes can help with all of that. Who do we call if we need help? We talked about our innate, the Koreans. Our innate, we know what all these guys do, and then special forces. Remember, special forces are very, very what? Specific to a particular antigen. Whereas your police force, your innate immune response will respond to anything. And we know, what is the one that's most prolific up here? Neutrophils. Neutrophils neutralize. Okay? What were macrophages before they were macrophages? Monocytes, smaller so they could get out and they mature. They're more long lasting. They eat a lot. Basophils are just like mast cells. Basophils are in the blood. Mast cells are in the tissue fluid. What do they secrete? Histamine. What does histamine cause? Vasodilation, right? To make your capillaries have more space between your endothelium cells so fluids can get out, causes swelling, right? And other cells can get out as well. Eosinophils eat the worm, go for the splinters. Natural killer cells, right? They can bind with a pathogen and then do like a lysosome-like action to destroy it or release a chemical to destroy it. Okay, so yes. Okay, we already talked about them. I'm just pulling this out in case you're reviewing and you want to see it. You could freeze it right now. Okay, we talked about the inflammatory response. I feel protective proteins. We talked about interleukins and interferons. Interleukins, what do interleukins do? Do you remember? Do I have anything up there about interleukins? What? Yes, yes, that's like Red Bull. Interferons are trying to do what? Warning. Yeah, it's a warning. And it says, I'm a cell infected by a virus, and you release a warning. It's like a fire alarm. It tells everybody else, be very careful because viruses are trying to get in your cells. Okay. All right. Complement proteins can do many things. One of the things they can do is drill holes to break the integrity of that pathogen that's invading in you. So and we know about natural killer cells. We know about antigen presentation cells, and they're very specific with our inappropriate cartoon. A helper T cell gets activated, and then it has to confer with a helper B cell that recognizes the exact same antigen. Plasma cells secrete antibodies. What are memory cells?
For next time.
For next time. Hopefully you have memory cells right now for polio in your body because you got a polio vaccine. Hopefully you have memory cells right now for measles, right? There's a measles epidemic starting all over again. So you want to make sure you have your MMR, right? Because if you don't have memory cells, then you can end up getting the measles, right? Anti-vaxxers, time to get vaccinated. Hurry, okay? And then macrophages like to eat. We went over this, right? You can freeze it on here if you need to review this some more, yeah? You want me to talk about it? I will. Okay, I'll talk about anything. Oh, super cool that our antibodies have two arms. Why? They can bind to two antigens at a time. The base determines how you'll be destroyed. Maybe you'll be engulfed. Maybe the base is going to fit into a mast cell. Maybe it's going to trigger a complement system. MHC1 is MHC2. MHC1, when you display the antigen, stabilized by a CD8 because you're going to get 8. MHC2s stabilize CD4. I am for you, not against you. When I say stabilized, who is that protein, stabilizing protein, coming from? T cell. T cell. Okay. We know all of that. I'll let you freeze it if you want to go over it some more. This is a good review if you want to go over that some more. We have reviewed that. Again, you could freeze that. Okay, innate, what you're born with. Acquired, you can get it passively or actively. If it's actively, you get to keep it forever. If you are somebody who is an anti-vaxxer and you're like, oh my gosh, I wish I was vaccinated, everybody in my family has measles, now I have measles, or I could get measles. What you can go get is a gamma globulin shot of antibodies that will attack the measles, and so you will be protected from it. So that would be artificial, and you could just get a gamma globulin shot, or you could begin nursing, but that might be awkward. Okay. The second exposure that you have, I'm tired. The second exposure that you have is much, much greater than the first exposure that you have to that particular antigen. Some shots require boosters. In order to really get the protection you need to get a booster shot of that particular vaccine to keep your antibody level high. Plant defenses, we talked about this, spines, leathery leaves, poisons, you could force it to go through. It's like, what time is it?
3:07.
Look, we could finish this one. You want to? I always tell you that.
I think we can.
You want to do it?
Yes.
I'll do it. Then we can be done with Big Idea too.
Let's do it. Let's do it.
Yeah, I have a meeting at 3:30, so I can keep
Yes.
going.
We can focus.
You can focus. I'll help you. I'll be better.
I'll help you.
I'll be better. Okay, 2E, ready? Many biological processes involved in growth, reproduction, and homeostasis include temporal aspects. Let's see what those might be. Huh, look at this growth. Look, where's Miss Litton? She's getting there, right there, okay? Reproduction, we know about that. It's all in the timing. Homeostasis, yay. What is apoptosis?
Cellular suicide.
That's right. Yeah, self-suicide. Why would you need to die? I know, your DNA has too many mutations in it. Maybe you're infected with a virus. Maybe we need to make digits, fingers. Maybe your time is just but that cell needs to die. Oh, external fertilization. Now, external fertilization, this means, I just didn't see that coming. Yeah, neither did she. Now, what? Now, okay, it's gonna work great. Focus. Okay, so on the frogs, they have to have a lot of sperm and a lot of eggs because they don't have it centrified in just one was centrified. I don't believe that's a word. Is that a word?
Centrified?
Yeah. You don't have just one location where that fertilization is going to take place. Whereas internal fertilization, I have teachers, we have a meeting at 3:30.
Wait, what's the difference between external?
Okay, external and internal fertilization. Somebody has a penis.
On the outside?
What? Yeah, you can get fertilized on the outside. Not you. But I'm taping this for YouTube, just so you know. That was Mrs. Heller. Okay. All right. You have to generate more eggs and more sperm for external fertilization. Why is that? Because it's not an enclosed environment. You have less control over that, right?
What is it like that?
Oh, okay, yes. Let me explain this. In external fertilization, the female is releasing eggs and the male is releasing sperm to fertilize them externally. Does that make sense? So that's external fertilization. Internal fertilization usually involves a penis. And that penis, its sole purpose is to get the sperm closer to the egg. Exactly. That would be internal. For spiders, it's like their back right leg is their penis. So yeah. And then octopus, one of the 8 tentacles is actually used to insert semen into the penis.
No.
Watch out for that 8th tentacle. Okay.
You know that?
No. Oh my God.
So this is a baby being born, a placental mammal. Yes.
Did you say that to the unicorn?
Okay. We're just. I don't think we need to know that anymore. Yeah, we don't need to go over that anymore because that's. Yeah, no, we don't have to go into the details of the reproductive system anymore. That's not part of the test anymore, so I don't have to go over that with you. But we talked about how ovulation occurs. Yes, do you remember this? Okay, so now look at these same hormones that we're going to use in spermatogenesis. So FSH, follicle stimulating hormone, and just instead of stimulating a follicle in the ovary, this time you're going to get your seminiferous tubules to produce sperm. Luteinizing hormone triggers testosterone. When you have a rise in both of these are going to exert what? Negative feedback, exactly. Okay, hypothalamus secretes gonadotropic releasing hormones. We talked about FSH and LH. We just reviewed that, right? Stimulating FSH triggers the follicle to mature and start releasing estrogen. That estrogen will cause a buildup in the uterine lining, and it exerts positive feedback. As the estrogen level rises, so too more GnRH, more FSH, until then it triggers ovulation, and at that point you have luteinizing hormone, and the follicle will burst, the egg will be released, and it becomes, due to luteinizing hormone, the corpus luteum, a yellow goopy mass, which will secrete progesterone in order to maintain the uterine lining. And we've reviewed that. I think we reviewed that already didn't we review that yesterday?
Yeah, we did.
Yeah, we did. Okay, so we already did that. Egg is popping. We already know this. That's easy peasy. Okay, totipotent. A totipotent cell has full developmental potential. So when you have one egg getting fertilized by one sperm in a human, it can divide a few times, and you could separate all those out, maybe 8 cells, and have 8 identical octuplets from that. Okay, that's a full developmental potential. But at some point, it loses totipotency. Like for sea urchins, it's after the very first division, the cells are already determined as to what they're going to be. Now, what we know is that the DNA is the same in every one of your cells. What's different about your cells? What's in the cytoplasm? Thank you. What's in the cytoplasm? Okay. Remember when we talked about morphogens, which control morphogenesis? And so what you have in your cytoplasm can then trigger the development of that particular cell. So we talked about where the sperm fertilizes the egg. That's where the anterior region develops. The opposite side is going to be where the posterior region, where you have the most yolk is the ventral region, and the least amount of yolk is the dorsal region. We call the front part the vegetal pole, the ventral part, not vegetable, but vegetal, and then animal pole is in the back. But we're so close. Morphogenesis, oldest bio buddy, tell the youngest bio buddy what morphogenesis is. Hurry, hurry, hurry.
And form the plasma. Go. Thank you.
You're welcome, sweetheart. Do you remember when we talk about when one cell induces another? And what they'll do is they'll induce it with morphogens, which are going to be turning on genes. What do we know turns on genes that we learned about in eukaryotic cells? RNA polymerase binds to the promoter. What helps RNA polymerase to bind to the promoter? Transcription factors. Perfect. So it's usually an inducer or morphogen is some sort of transcription factors that will influence that. We're not meeting till 3:30, right? 3:30. Okay, good. And we talked about a solid ball of cells is called a marula, and you pump in some salt. Who follows it? And you form a blastula. For humans, that's on day 5. And then that's a hollow ball of cells. Gastrulation, that infolding, right, forms a layered ball of cells. At first, you just have endoderm and ectoderm. Later, mesoderm. From the ectoderm, what do we get from the ectoderm? Skin and your eyes and your nervous system. From the endoderm, you have
digestive
and respiratory systems, any organs associated with it in the tube, and then everything else like muscles and bones comes from the mesoderm. Okay. Now, for us, once we form a blastula, inside there's called an inner cell mass, and that inner cell mass is what actually develops into the human. So when we talk about things being totipotent, like the marula, it has full developmental potential. When we talk about pluripotent, that's that inner cell mass gives rise to all the cells of the embryo. Unipotent, what does that sound like? One. Unipotent is you've been specific enough, become determined, and you already started differentiating, and you can only be one type of tissue. Okay, you don't need to know all that part. That's scary. We've already talked about that. Induction, we talked about how we activate genes with transcription factors and morphogens. We did homeotic genes. Uh-oh, bad fly. Look at that, antenna coming out his eye. We talked about genes that are conserved, right, that have to do with control. And what? Yeah. sorry, I need to breathe. Hox genes we talked about. If we look at the order of them, we can see that similarity. It doesn't matter if you're a fruit fly or a mouse or an embryo. What percent of your genes do you share? And quite a bit, right? There's only about maybe a 1% difference between us and, like, say, another primate. And that's that. You don't need to know that anymore. Have a baby, just don't do that. Transpiration, that's responding to your environment as well for growth and development. TATC theory, let's go over what those stand for. T is transpiration, then what? Adhesion, which is what? Adhering to the xylem walls. Cohesion. Oh, I just told it to you. And TAT, what was the one I didn't give you? Tension. And that's going the water potential gradient, yes? Are we there? Stomata, open and close. Are we getting close? We're getting super close. Thigmotropism, do you remember what that is? When you, like, wrap around an object, you're responding to your environment so you can climb and get support. Stinging cells. Morphogenesis. I don't think I gave you the definition, but this is when the whole plant responds to, like, a windy environment up on a hill. Nastic movements, like when the praying plants where they close their leaves. Communication. Tactile. Do you remember the bee dance? We already reviewed that. Cyclins. Do you remember cyclins and kinases and how they need to be? Yeah, the cell cycle. Good. Okay. External. Did we review the cell cycle yet? Okay, good. You're like, yeah, we did. Just stop. Apoptosis, we did that. Telomerase activity, that's a good one. On origins of cancers, normally your chromosomes shorten every division, but if you have cancer, they're continuing to stay elongated. We talked about the two genes, proto-oncogenes and oncogenes. Didn't we already talk about that? I'm looking at Alan because he's been at every single one. Proto-oncogenes, remember, these are the ones that say go cell cycle, go, whereas tumor suppressor genes are saying, hey, we need to commit apoptosis because we have a problem. Can you remember an example of a proto-oncogene? It's like RAS is like stepping on the gas. And then what was an example of a tumor suppressor? P53, good. We reviewed that. Pineal gland. Do you remember melatonin? That's responding to your environment as well. Melatonin level does what when you sleep? Rises. And then light will make it fall. Good. Okay, yeah. Behavior, homeostasis, metabolism. I think ultimate source of all energy is the sun. Do you know the difference between hormones and pheromones?
Yes.
Pheromones are at a distance between two individuals, and hormones are at a distance between two different body parts. Good job. Responding to stimuli, reproduce. Binary fission, yes? That's what your bacteria are doing right now. Oh, do you know why I have this here? Countercurrent exchange in his nose to recover moisture in his blood supply. That's responding to your environment. You grow, develop, sperm, egg, making new. Natural selection is the what for evolution? Can you give me one word? Mechanism. I don't know if that's going to say that, but yes, it is. Mechanism for evolution, right? Are there other mechanisms for evolution besides natural selection?
Yes.
Tell me. That would be adaptation. What else? Small population size, non-randomating, mutations, gene flow. Good. That's it. We're done with big idea 2. Way to hang in there. Okay, done. Big idea 2. So now we have big idea 3 and big idea 4. And we have Wednesday, Thursday, and Friday to review those 2 big ideas. That's the hugest one. It had 240 slides in that presentation. Yes, I'm super proud of you. Have a piece of toast. Review.